Hello everyone. Hope all of you are doing well. Now today I will discuss the second part of the topic, the remaining part of overview of bacterial endospore. So in the previous video I have discussed about the definition of sporulation, sporulation in bacteria, the process, the definition of sporulation, and spore forming bacteria list, features of spore forming bacteria, and about structures, resistance mechanism of endospores, and the sporulation process in details. So today we will start our discussion from germination. That is the opposite of sporulation, as the return of Captain America. Means when conditions improve, the dormant spore wakes up just like Captain America coming out of the ice. It thaws out, absorbs water, and goes back to saving the microbial world as a fully active vegetative cell. So what is the germination process? Whenever favorable conditions return, the spore again germinates. That is, it enters into metabolically active phase again from the metabolically inert, dormant, resistant spore. So first is the activation. Physical or chemical signals such as heat shock, nutrient availability like amino acid, sugars, or pH changes, whatever be the favorable condition, those activate germination. Germination means enzymes degrade the cortex, increasing the water uptake. So, what we have learned in the previous video of sporulation, the cortex with less water content in the structure part. We have seen that the core and the cortex both have the less water content, low water content and low water activity. So here enzymes degrade the cortex, the outside layer of the core, and increases the water uptake. Degradation of SASP again releasing the DNA. SASP means small acid soluble protein of spore, and it releases the DNA. And outgrowth, cellular structures such as membrane, ribosomes get dehydrated. Metabolism resumes, forming a vegetative cell. So these are the initial steps of germination. Forming the vegetative bacterial cell, which is metabolically active. So three steps are involved in germination: activation, germination proper, and then outgrowth. So coming to the molecular regulation, SPO0A as the zero stage spore forming protein. This is the mastermind regulator. So this is the Tony Stark of sporulation. Activated SPO0A represses the vegetative growth genes and initiates sporulation specific genes. It regulates downstream sigma factors. Sigma factors are the sporulation genes. So it regulates the downstream sigma factor. Stage 0 sporulation in the gene or enzyme producing factor is the SPO0A. So it is the mastermind regulator. So what is the sigma factor cascade? That is the movie director crew. Sigma F, it handles the early stage setup like scouting locations, etc. Its actions are like these. Activated infospore regulates the early sporulation genes. Sigma E, these are involved in costume designing, means they are active in the mother cells, regulates engulfment and coat synthesis, the next step. And then comes the special effect team. Special effects team, that is the sigma G. Those are the final regulator in the mother cell for coat maturation, spore coat maturation. Then sigma K, they do the final editing portion, ensures everything looks perfect for the release. Okay. Then other regulators, GER proteins, those are the spore germination receptors which detect specific nutrients, whether they are present or not. If present, then switch to germination. If not present, then switch to sporulation. SPO3A or SPO3E, they mediate four-spore mother cell communication because engulfment occurs when the four-spore is present along with the mother cell due to asymmetric division. Two-component system of environmental sensing and signaling, that is the KEEN A, SPO0F and SPO0B, which we have learned in the sporulation process in the initiation phase, which phosphorylate SPO0A, this two-component signaling system, and causes the sporulation process, starts the sporulation process. Then what are the clinical and environmental importance of this sporulation or the endospores? Medical challenges, Clostridium difficile endospores resist common disinfectants, complicating hospital infection control. Whenever there is use of antibiotics for longer duration, more hospital stay, prolonged hospital stay, then there will be Clostridium difficile infection, Clostridium difficile diarrhea, because they resist common disinfectants. So they complicate hospital infection control. Bacillus anthracis endospores are stable in the environment and cause anthrax. Clostridium botulinum. So they are the threat for us, for human beings, because they are resistant to disinfectants, common disinfectants. Clostridium botulinum spore survival in improperly canned foods leads to botulism. In food industry, why they are challenging? Because endospore forming bacteria like Bacillus species are major contaminants in dairy and processed foods. Control measures include autoclaving, control measures including autoclaving, high pressure processing and irradiation. Those are the control measures. Biodefense research on rapid detection and neutralization of endospores, as for example, anthrax bioterrorism agenda is ongoing. Now, what are the laboratory techniques? Staining by Schoffer-Fulton method. Usual staining, we can't see the endospores. Gram stain or normal Zn stain, we can't see them. Malachite green stains spores, safranin stains the vegetative cells. Electron microscopy, the Schoffer-Fulton stain method has two types of staining together, malachite green as well as safranin. Malachite green stains the spores and safranin stains the vegetative cells. Electron microscopy reveals the structural details. And molecular tools, PCR, detects spore-specific genes such as SPO0A and GERA, and proteomic analysis identifies resistance-associated proteins. Heat resistance testing exposes spores to 80 degrees centigrade for 10 minutes. Surviving spores confirm true resistance, whether the process is resistant to spore or not. So, if the spores are present, that means they are resistant to spore. The surviving spores confirm true resistance. Now, recent research directions. Mechanistic insights, advanced imaging, as for example, cryo-EM reveals spore coat organization, cryo-electron microscopy. Studies on DPA, dipichlorinic acid biosynthesis and regulations can be done on endospores. Spore surface engineering, functionalized spores as drug delivery vehicles or environmental biosensors. Functionalized spores can be used as drug delivery vehicles or environmental biosensors. And genomic insights, comparative genomics to identify conserved sporulation pathways across genera, different genera. Discovery of non-canonical spore-forming bacteria outside Bacillus and Clostridium. So those researches are ongoing. Now, that's all about description, the resistance mechanism, molecular regulation, recent researches on spores. Now, now some questions. What chemical in endospores helps stabilize the DNA? That is the obviously dipicolinic acid. It is like the supply of canned food and it stabilizes the DNA. Which bacteria would survive a zombie apocalypse? That is the Bacillus subtilis, because due to its power to form indestructible spores. Now, what is the first step in sporulation? First step in sporulation is when signals come, it is the asymmetric cell division. Asymmetric cell division is the first step of sporulation. And you can revise. Then what is the second step? That is the engulfment of spore by the mother cell. Asymmetric cell division means formation of mother cell and spore, and then spore will be engulfed by the mother cell. Next step is the cortex formation. Initial stage is the signal induction, but this option is not available. So first step is asymmetric cell division, then spore formation or then engulfment, and then cortex formation, followed by spore coat formation, maturation, and finally the release. Then what does the spore coat do? Spore coat gives the protection against harsh conditions. So you can revise the structure while studying this question. Spore coat, it protects from enzymatic destruction, as well as destruction by chemical substances. Enzymatic destruction protects from protease and lysozymes, and chemical resistance due to permeability, as it is impermeable to toxic agents. So those are the references. And then some fun with endospore. Why did the endospore refuse to party? Because it was in dormant mode. And what is the bacterium's favorite vacation spot? The culture club, because all the materials are available there. They are metabolically active there in culture media. Why don't bacteria ever get lonely? Because they are always dividing their time, always dividing their time. How did the spore feel during germination? They imbibes water, because in spore there is low water content and in vegetative cell water content is much more. So they feel like, oh, I am swelling with excitement. Now, what is an endospore's motto? What doesn't kill me makes me stronger, literally. So no one can kill me. That is the endospore's motto. Why don't bacteria do well in relationships? Because they're always growing apart. Why did the microbiologist go broke? Because they lost their culture. How do bacteria communicate at parties? Through cellular networks. And what is the bacterium's favorite superhero? Spore man. Definitely, it's a superhero because no one can distract it. Why was the petri dish so good at telling stories? Because it had a lot of culture. So, hope you enjoyed the session. Don't forget to like, share, and subscribe. Thank you. Have a good day.